So this is a transplant study for patients with a peripheral T-cell lymphoma, and it's an autologous stem cell transplant study. And generally, the standard of care for these patients is that if they achieve a remission with the initial chemotherapy, they undergo, if they're eligible and young enough and healthy enough to undergo an autologous stem cell transplant using chemotherapy as conditioning regimen, which is, in most cases, the standard is BEAM-based chemotherapy. It's high-dose chemotherapy. But we have recognized that this is not a curative therapy, that most patients who undergo this process will eventually relapse with their disease. So we have designed this trial to sort of beef up the initial conditioning regimen so we can have longer, more improve long-term results in terms of survival and progression-free survival. That's the idea behind it. So this is designed as a phase 1 study using an antibody that is directed against CD25, basiliximab, and it has a radioimmunotherapy target or radiation target attached to it with the help of a chelation process where yttrium-90 is attached to the antibody, and the antibody then targets cells that express CD25, and the radiation is delivered directly to the cancer cells, and that provides an added therapeutic modality for these patients. And that was kind of the whole idea of trying this, or designing this trial. So this antibody, the basiliximab, as I said, targets CD25, and basiliximab itself is approved for various forms of immunosuppression, particularly in organ rejection, because it depletes T cells. And in malignant T cells, in peripheral T-cell lymphomas, we have seen overexpression of CD25. So this seems to be a good target and a good sort of antibody to use for this particular process. So that's the background of the study. And this is because everything has to start with safety analysis, we need to make sure things are safe for the patients. So this study is a phase 1 study, and we are looking at 3 different dose levels of radiation to be delivered to the patients through this radio-labeled antibody in combination with standard dose BEAM chemotherapy in the setting of transplant. So it's a 2-step process. The first step is to establish dosing of the agent, or what's called dosimetry in nuclear medicine. So that's really not my area, but my colleagues have helped me with this. So you need to know the distribution of the radioactivity that you're going to be giving to the patient. So make sure it is not untoward, it's not something that's undesirable, or for example, if it concentrates in the lung or the kidney, it could be toxic to the patient. So the first step is using an indium-labeled basiliximab antibody. Now, indium is a gamma emitter, and it doesn't provide any therapeutic benefit other than the antibody binding to CD25. It doesn't provide actual radiation to the tumor, but it is, since it's a gamma emitter, you can use X-rays to establish its distribution in the body. So at day minus 21, if you look at the study design, they receive an injection of this indium-labeled basiliximab at 5 millicuries. It's a standard dose. And after that, they undergo a series of SPECT scans using X-rays, X-ray equipment, get scans at various time points, 24 hours, 48 hours, and up to 144 hours to see where this antibody and its radio labeling is being distributed in the body. And if the distribution is acceptable, the patient will then undergo therapy. And the therapy will start at day minus 14, where they will receive an injection of the yttrium-labeled, or the hot antibody. We have tested 3 different dose levels, starting at 0.4, going up to 0.5, and then 0.6, which is our target dose. Since this was a safety study, we were going to look for safety signals in each dose cohort. So they get their dose, and then they get admitted 2 weeks later for their BEAM transplant. And based on that, then they undergo through the whole stem cell transplant process as standard of care. And then we looked at their additional toxicities that this process may have added. Because transplant generally is a toxic process anyways, we have used special criteria called Biermann's toxicity criteria that have been established toxicity criteria for looking at any additional toxicities besides or outside the standard accepted toxicities for patients undergoing transplant. So that was our, that's what the process we use for evaluation of toxicity. And we've so far been lucky enough to not have anything that was dose defining or dose limiting in terms of the dose escalation of this radio-labeled antibody. And so we are now at our dose, the highest dose level. We have enrolled 13 patients, and out of which everybody engrafted on time except one patient. The first one had a delayed engraftment of her platelets, which is why we expanded the cohort from 3 to 4 patients. And subsequently, there have been no problems in engraftment. There have been no excessive incidences of mucositis, which is the other main toxicity of transplant. And we have really not encountered any dose-limiting toxicity according to the Bierman criteria. The patients, of course, had their usual accepted transplant-related issues. So we feel very strongly that this dose is safe. This trial has not been designed to test efficacy, but we will expand it to include another 7 patients to get an efficacy signal. In terms of outcomes for the disease, the median follow-up on patients that there's a trial still ongoing is about 10.6 months, and out of which 3 patients have relapsed. And 2 of them have passed away from disease progression, and the third one just relapsed and is undergoing therapy. So again, the trial was not designed for efficacy, but people who do this feel that this is quite encouraging. A 10.6 median follow-up for most of these patients without disease seems to be encouraging. So we'll see what happens as the dose, as this trial expands, and we'll see if we find a signal that is worth taking this forward into a bigger trial. Because if this works, then we may have another option for the patients when they undergo transplant.